everyone, and thank you for joining my talk. This is a new project, and it is the result of a collaboration between the Crop Functional Genomics group of Franco Holdinger in Bonn and the LibCa lab in Urbana-Champaign. And the current working title is How well do GWAS methods perform on TWAS data? And what GWAS methods and TWAS data is, I will explain right now. Plant breeders and also researchers are interested in finding genotype-phenotype associations. So we want to find a position or a gene in the genome where we can see that a different information at this position increases, for example, plant height or has any other phenotypic effect. And for that, we usually need a diverse population that has variants for a specific trait. And then we analyze the genotypes. We can test for an association between the genotype and the phenotype at a single marker and record the p-value. And if we do that for all the markers that we see across the genome, then we get a plot like the one on the right side. And we can identify novel associations between genotype and phenotype. There's a variety of mathematical models and tools available to do this for this type of data. Since a little more recently, we also got large-scale transcriptomic data as the intermediate step between the genome and the phenotype. However, gene expression is not only influenced by various genetic loci, but also by the environment. So what we get is continuous data, but in this data we could also observe gene expression that is correlated with a trait variation. To find associations between expression data and the phenotype, people actually are sometimes using the same tools that we use for the GWAS analysis. However, the GWAS analysis was developed on different data. And so here are some of the differences between expression and genomic data. And so, for example, marker data, as we've seen, is usually discrete, but expression data, as we can see here, is on a continuous scale. At a similar level, markers can be linked, so they can be closely together on the genome. However, in gene expression, we can have correlated genes, so that if expression of one gene is strongly correlated with another gene or with a huge amount of other genes. In addition, what complicates this analysis is that expression data can also follow different distributions, so we might not necessarily have a normal distribution, but maybe two peaks or something. And as I already mentioned, there's a range of different models and tools available for these analyses. So what we want to find out in our study is how well can we identify association between gene expression and the phenotype using GWAS methods. And for that, we are simulating phenotypic values in a way that we know with which genes they are associated, so that we have like a ground truth that we can test for. And then we will use these generated data sets to perform the TWAS analysis with different tools to see how many genes we can identify. We also want to use different genes with different properties. So as I said, genes, for example, that are highly correlated with a number of genes versus those that are not. And we would also like to test different populations. And then we would like to see which factors influence the quality of the detection. So the questions that we hope to answer in more detail are: what can we expect when running a TWAS? So what are the limits in terms of the number of genes or the heritability? So how many genes will we even identify with decreasing heritability, for example? We want to find out if there are differences when we use different data sets, for example, a biparental population or a diversity panel. And we also want to see if the false positives or the detection rates change depending on the properties of the genes. So for example, their distribution or their correlations. And then we would like to find out if there are strengths for different models that we can use for further analyses. And then if we still have some time, we might want to find out how GWAS and TWAS complement each other and extend this TWAS analysis with some genomic predicted data. Yeah, with that, I would like to thank you all for listening. I would like to thank the LibCa Lab especially, but also my group in Bonn and also our funders. Thank you very much, and I will be happy to answer your questions.